Professor Alev is both the chair and a professor of psychology at the University of Virginia. She aims to understand how axon reorganization, synaptic pruning, and sensory plasticity occurs through identifying the molecular involvement, and her research also involves animal models. So please, without further ado, I would like to welcome her again and start this session. Thank you.
Thank you so much, Didar, for this beautiful introduction. And also, thank you so much for inviting me to this festival. Let me try sharing my screen and continue from there. It's great. It is really a great pleasure to be here and be able to talk to you. I wish I could be in Sarıyer with you right now. I spent several weeks at Koç University many years ago, and I still remember the beautiful view, which I think I'm seeing at the background of many of your profile pictures now. Please feel free to stop me anytime, ask questions, and I really like this to be interactive, or if you want to ask questions afterwards and anything on science and life and careers, I'll be happy to talk to any of you. So to give you a little bit of background about myself, I graduated from Chapa University many years ago and moved to United States to pursue my PhD in behavioral neuroscience. At the time, there were actually a total of 0 such programs in Turkey. So I'm really pleased to see how neuroscience training has thrived since then. Throughout my research career, I worked with animal models. The first animal model I encountered at my PhD work at SUNY Stony Brook was tree shrews, this animal that you see here. And I'm going to mention that later on. Then I switched labs and started working with cats and monkeys. And I completed my dissertation studying the anatomy of the synaptic circuitry in a brain nucleus along visual pathways, that is the LGN, lateral geniculate nucleus. During my postdoctoral fellowship years in NYU, New York, I worked on ferret and mouse sensory cortex, both at the anatomical and the cellular level. After I opened my own lab in Virginia, I continued to work with ferrets, mouse, sometimes prairie voles, and rats. And then examine the development and plasticity of, again, the sensory pathways. So today I will tell you about some of these studies in the context of the advantages and caveats of using animal models in basic science. Sensory systems are great models indeed to understand the brain circuits and cellular function. This is because first, the beginning point of the processing stream for perceiving a physical stimulus, that is where the receptors for the physical stimuli are, is very obvious. It is either the eye, the nose, the ear, the skin, so very accessible. This is in contrast to some other brain functions, such as memory, fear, sadness, hunger, where it is very difficult to figure out which brain regions to stimulate or starting the chain of actions that lead to those emotions and behaviors. Then the physical sensor stimulus is also very easy to generate and parse into its components, its features that we actually perceive. For example, for vision, these features include colors, change of light, contrast, edges, depth, movement. For taste, it's sweet, salty, sour, bitter, and umami, the taste of the proteins. In addition, the cellular, molecular, and genetic mechanisms of brain development and the plasticity seems to be common across the entire brain. These many systems share common mechanisms. So sensory pathways become this very easily accessible places to study these mechanisms. So how can we study how the brain generates the perception of the physical world around us? Human psychophysics, there are many tools available for us to study human. Psychophysics, lesion analysis, neurology, brain imaging, cognitive neuroscience. We have a lot of tools at hand for working on human brain and to decipher the features of the percepts. But there are also gross anatomy, and which we use for how sensory pathways are embedded in the brain. And we have a general understanding of how the nurture, the environment can shape the brain. However, the questions that require these currently invasive approaches to understand the molecular and genetic players in synaptic transmission, development, and adaptation, we go to animal models, which invariably use smaller, similar mechanisms to sense and react to the external world as we humans do. So this is evident in the phylogenetic relationships for the major subclasses of mammals. That's that I show in this slide. That this one here illustrates that all species that are examined had a constellation of cortical fields that includes primary somatosensory, visual, and auditory areas, although their relative size and the locations have been altered in different species. Even though we learn from a fly or a lizard eye, but the things that we learn from these species, which are really phylogenetically very remote species for us, inform us about how the sensory stimulus is detected in the human eye. And there are also variations in the strategies different species utilize as input for more complex behaviors, such as aggression, as in this example. And they can correlate with the differences in sensory pathways and function in these species. In this example, olfaction rather than the vision or hearing is the main sense for rodents to detect a predator or prey, which explains the disproportionately well-developed olfactory bulb in these species in rodents. So here's an example of how easy it is to decipher and generate the sensory stimuli. When you look at a visual scene like this, our visual system discriminates the scene in features. It detects the contrast, it detects the color, it detects the movement, even though it is a static picture, and it detects the depth. Because when we look at it, we get to see that there are seagulls a lot larger here, and the seagulls a lot larger there, and this can only happen in an environment when there is a depth 3D image. So let's talk about that a little bit. So we are seeing things that are gradually farther away from us because we know that the objects that should have the same size look larger when they are closer to us. This is actually comes in handy, and sometimes it breaks down depending on the tools that we use. A more reliable tool or the cue that the humans and other species that have their eyes in front of their face, the frontal eyes, the cue is ocular disparity for depth perception. So a simple demonstration is something what you can do anytime. If you put your finger up in front of you and fixate on it while you're looking at the objects away from your finger, and look at it with one eye first and then the other eye. What you see is that the scene behind your finger moves, or your finger jumps from one side to another, depending on which eye you are looking with. This is because two eyes capture a slightly different view of the visual field, and it's called the ocular disparity. Ocular disparity is the basis of many technologies that are developed to make us perceive the depth. And the three-dimensionality to make us fool into thinking that there are three-dimensional objects in front of us just by looking at, even though we are looking at two-dimensional images. We know that the ocular disparity is a function of how the sensory pathways are organized in the brain of the animals that have frontal eyes. The central visual field is depicted here. Is encoded by both retinas owing to this cup-shaped structure of the eyes. The axons of the retinal ganglion cells get to be reorganized, shuffled back so that they sort the axons that encode the contralateral hemifield in the hemisphere of the brain and on the other side, on that side. So when those inputs terminate in the lateral geniculate nucleus on separate layers, carrying a multiple maps of the contralateral visual field as seen by the one eye or the other. And when these axons, the axons of the relay cells in the lateral geniculate, when they project to the striate cortex, they have actually make a single map. All of these axons become interdigitated so that they merge their maps without merging their inputs. And the inputs from the ipsilateral and the contralateral lamina, small visual fields that they represent, gets to be represented in these different columns in layer 4 of the visual cortex. As a result, the cells in the two adjacent columns have slightly shifted receptive fields, and they carry this when they project to the upper layers in the cortex. So now there is a milestone in the neuroscience history, which brought the sensory systems at the forefront of the studies to understand the cellular and molecular mechanisms of the brain function. So this I'm talking about the work of the Hubel and Wiesel in the 1960s, who applied the approaches that were available, developed by them independently, electrophysiology and cellular intracellular recordings. And they studied how the neurons in the cat visual cortex responded to light. And they identified the different cell categories that respond to spots and edges and moving stimuli and so on. And after many studies that culminated to a Nobel Prize for them and the work of the many other neurophysiologists and neuroanatomists, revealed that how the information can be coded and processed in the visual pathways of various species. The vision became a template to start understanding other systems and the brain in general. So we now know about the pathways and parallel pathways of the feature processing of the visual system. We know about the WER pathway and the WAP pathway in the cortex. We know their response properties of these different cell types. We know the neuronal connectivity in sensory pathways, very complex connectivity, feedback and feed-forward connections that occur throughout the cortex and in the subcortical structures. And more importantly, we know that, more important to me, that we know that the inputs and outs of each nucleus here that make these nodes and the type of inputs that come onto them and the neurotransmitters and receptors utilized by those connections. And all of these become the accumulated knowledge. We also know that there are variations of these characteristics in different species. So there are similarities and differences in how sensory pathways are organized in different species. And for example, well, these differences can allow us to tease out what features are relevant for the survival or the day-to-day operations of the particular species or animal species. For example, what I'm showing you here is the anatomy of the lateral geniculate nucleus in many species in the primates, and the pre-primates, carnivores, and rodents. What we get strikes us is this laminar organization. This is where the eye and feature-specific inputs are segregated. And this holds true for many higher mammals in the primates, in the humans, and the tissue. On the other hand, look at mouse. Mouse algianis right here. There is no discernible lamination or segregation in that. A mouse is a species that gives us many opportunities due to their suitability for transgenic technologies. It has a rather differentiated algae, and it does not have any binocular vision. It does not have ocular dominance powers. Yet, at the cellular level, many mechanisms that are found in mice in terms of the neurotransmitters, receptors, or postsynaptic second messenger system level, many mechanisms that are found nicely apply to those in the higher mammals. So it becomes important to identify the common organizational principles of sensory pathways and the differences across species at the anatomical and cellular levels. So the focus of my lab's research is right at that intersection. We aim to reveal the synaptic circuits in sensory nuclei, particularly in the thalamic nuclei and the cortex, primary sensory cortex. And we use available animal models that may have the most overlap with the human behavior or the development, depending on the feature that we're looking at. To understand the circuitry, the first thing one needs to do is to understand this anatomy. And as a neuroanatomist, this is actually, I like saying that. Yeah, unless you know the anatomy, you're not going to understand the function. So that means that we need to know the neuron types in each nuclei and the synaptic inputs that drives on those individual neurons. We need to know the intrinsic and modulatory synapses that modulate the neuron's excitability. We need to know the outputs, that is where the information processed in those cells take into next. To know these and understand these, we use many anatomical techniques, including light and electron microscopy, track tracing, immunostaining, and quantitative modeling approaches. So identifying the cell morphology has fascinated many early neuroanatomists like Prof, who over 100 years ago used the Golgi technique to illustrate the neuronal morphology across many systems and species. And looking at these drawings, Cahal and others also envisioned that communication between neurons must have taken place at a specialized structure, which they called synapse at the time, but they did not have any evidence for it. The actual evidence for the presence of the synapses came after 1950s, when the electron microscope was invented. An electron microscope uses an electric beam rather than a light wave to refract or transmit through a thin section of brain. Because it is not limited by the wavelength of the light rays, electron microscope has the resolution power to visualize objects that are physically too small to be visualized, which is impossible to visualize on a light microscope. As a result, the electron microscope, despite all the other technological advances happened in the last 70 years, is still the only instrument that can confirm the presence of a synapse and the different organelles and its also relationship with each other. What are its limitations? Its limitation is that the material to be imaged at the EM has to be very thin, about 50 nanometer thin, and it takes a lot of tissue processing to be able to cut those. In addition, the electron refracting molecules that has to be metal molecules, they have to be attached to the cellular lipids, that is the biolipid membranes. This makes all cellular structures that are surrounded by lipid membranes visible. So the result is something like this, a monochrome image of a very thin cross-section through all brain structures. Here is what a neuron, cell body, and dendrite, and all sorts of other neural look like at electron microscope images. And these kind of images gave us the data that led to the textbook illustrations of the cellular neuronal structure, the organelles in them, and the connectivity, and so on. To give you a sense, these little gray bean-like structures here, they are mitochondria. And here is the nucleus, nuclear membrane, it's beautiful, chromatin inside it, and there's the cytoplasm of the cell, and the dendrite just emerges from it. What else? So here are some myelinated axons. The thicker dark surroundings are there because the myelin is made out of stacks of oligodendrocyte membranes that surround the axon. So, here's a slightly larger view. There's the mitochondria. Now you're probably able to see the cristae inside them. Here is the endoplasmic reticulum surrounding the nucleus. They are ready to catch the mRNA to start synthesizing proteins. What else? There are dendrites. There's a dendrite, there's a longitudinally cut dendrite, and this is an axially cut dendrite because they go in many different unpredictable orientations. Here's a synapse. Maybe here's another synapse. Let me show you a larger picture, very larger picture. So here's a synapse. These are vesicles. Here's a mitochondria, again, give you the scale. These are neurotransmitter containing vesicles. This is the presynaptic membrane. Right here is the synaptic cleft and the postsynaptic density. It's beautiful synapse. So we glean a lot of information from these images. Here's again another axially cut dendrite. You get to see the microtubules inside it and the mitochondria again. And we get to see a spine emerging from it. And that spine itself receives an excitatory synapse from this terminal. We know that it is an excitatory terminal because of this postsynaptic density. Compared to this one, how the postsynaptic density is thin, and the terminals like this one are usually GABAergic inhibitory terminals. So as my dissertation project in the 1990s, I characterize and quantify different inputs that impinge on the relay and interneuron cell populations of the visual thalamus. So identify those inputs by their morphology, in the reactivity for different neurotransmitter markers, and by track tracing. So here are some examples of those different terminal types. This is a vesicle field profile. That's called F here. It's an inhibitory terminal and synapsing on this relay cell dendrite cross-section. We know that this is an inhibitory, but this is not inhibitory. It's a relay cell, and this is not an inhibitory because we use the immunolabeling to mark the interneurons on these sections. And the antibody was packed with the colloidal gold, so all these little black dots you see are telling us that this is a GABAergic terminal. Also see a smaller excitatory terminal here, making another synapse on the dendrite. This big terminal here is coming from retina. I identify it as coming from retina because of its mitochondria, which is really pale. This kind of pale mitochondria is a very kind of a peculiar property of the retinal ganglion cell axons, and it's a really good marker for the retinal inputs. And this one is forming two synapses on a relay dendrite here. Let's see, on a relay dendrite here and onto an F terminal. which itself forms another synapse onto the same dendrite, forming a triad here. And there are glial membranes in pink here that surrounds this bundle of terminals here that makes this called into a structure that's called glomerulus. So the triads and the glomeruli are characteristics for not only to LGN, visual thalamus, but all sensory thalamic nuclei, allowing for, it's thought that this allows for temporal limited excitation to occur on the relay neurons, that retinal input can come in a really fast succession so that the inputs will not merge onto each other on the relays. So at that time, we also identified other modulatory inputs coming from brainstem and cortex by track tracing. A sample is here. The tracer is visualized by diaminobenzidine, which makes this dark precipitate in the terminals. We also use electron microscopy antibodies to identify each terminal and the origins of these terminals. This is an example of the polymethyl transferase labeled terminal, telling me that these inputs are coming from the brainstem. So the different cell types in the LGN receive a different composition of inputs. I skipped showing you the quantification graphs of the previous study, but this is, I just want to show you a summary of the gist of this specificity. When a cell type receives a different pattern of input than the other one, it is understood that the input processing, signal processing in these cells occur in a different way. And these can, for example, the X-type relay cells in the cat LGN receives this really glomeruli and the triads and very close to the relay cell body, and which is not a property of the Y-type cells in the cat. So we picked up on these studies many years later. There's a current study because a very nice technical development occurred in the last 10 years, and that allowed us to study the geniculate circuitry in 3D, three-dimensional reconstructions. So this is a flipbook video of images taken from the mouse LGN in 50 nanometer slices. It is prepared by a scanning block-based electron microscopy approach, which provides images of adjacent sections. And these sections come in perfect register to each other, which allows us to construct three-dimensional structure of the dendrites and terminals very reliably. Here's a neuron moved in, here's a nucleus again, and the cytoplasm, and all sorts of neuropil going on there. So we do the reconstructions by going through these images one by one. So identify a dendrite, here's a dendrite and a spine coming out of the dendrite here, and it's the colored version of it. And we follow that through adjacent sections until we capture its full 3D structure and the volume and other quantitative measures of it. In our project to identify the selectivity of different inputs on LGN dendrite segments, we first constructed the dendritic structure like this one that makes the fork branching. Then we identified all of the synapses in red here that form onto this dendrite. And then we constructed all of the terminals that make those synapses. Great. And then we could actually also sort the terminals, dendrite segments, whether or not they are primary coming out from a soma, or they kind of branches that come out from the primary or secondary branches. So the results I will show you are based on 26 such dendrite reconstructions and over 1,000 terminal glucons, some of which are actually mapped onto some of these dendrites. None of these dendrites show any morphology that is characteristics of interneurons, so we call them the relay cells. And now when we look at the volume frequency distribution of these terminals in this little graph here, we see a really large range of sizes to begin with. There are small ones and very large ones. Some of them probably coming from the cortex, and these are the retinal terminals. But the distribution is interesting because it shows multiple peaks. We know that the inputs coming from a single origin is actually make a very uniform type or size of terminal boutons. So their distribution of a unique input would be a normal distribution of them with single peak. When we see multiple peaks like that, we know that it is consistent with many terminal boutons originating from multiple distant sources. There is a way to actually further understand how many of different inputs are there. But before we can take advantage of those approaches to identify the distinct bouton classes, we had one more morphological criteria that we can use. And that is the mitochondria contrast, which I told you that is specific for the retinal inputs. So we did that. We separated boutons that have light mitochondria from those that have no mitochondria or just the large ones. And you can see the volume frequency distribution of these different subcategories here. One thing to notice here is that both the light mitochondria that we know that come from retina and all the others, a combination of cortex and the brainstem and the potential interneurons, still displayed multiple peaks. So great, we can handle that. There is a way, as I said, to figure out the unique subpopulation that can contribute to a multi-peak population. And that is called, it's a statistical modeling approach, and it's called the Bayesian information criterion analysis. And we follow that by a clustering analysis to find the peaks and the means and the cutoff points of these different populations. So I will spare you the details of these analyses for the benefit of time, but I will be really happy to talk about it if anyone is interested in these kind of statistical approaches. So this analysis here revealed us 3 different distinct retinal inputs: the smaller, medium size, and very large ones, which may correspond to different retinal ganglion cell axons, which we know that exist. We also were able to sort by the volume of the non-retinal terminals into subpopulations that are consistent with the very small cortical input, the blue ones, or from brainstem that's marked in the yellow, and the 3 different groups of inhibitory inputs from orange to red here. And when we apply these volume cutoffs to our data set, we were able to reveal the selectivity of different inputs to different segments of the relay dendritic group and potentially on different relay cell types. So here's a dendrite, and its boutons are constructed and color-coded for their size category. And here is an illustrative stick figure for what a dendrite looks like. Here, where we can see the dendritic structure and the composition of all bouton types that synapse on that dendrite segment. And these are all color-coded. And we were also able to sort the dendrite segments as putative X-type and non-X-type based on the presence of these grape-like appendages, and also the glomeruli that have been known to occur in X-type cells. And this is, here is it marked in purple here. And this is another example where the boutons are color-coded for the volume cluster categories. As we examine these, a pattern started to emerge. Like here, all retinal terminals are clustered on the secondary branch of this dendrite segment. Whereas the tertiary branches receive only the small cortical and some inhibitory signals. There were no retinal input coming onto these tertiary branches. And here are some more, standardized examples of this kind of a input segregation that occurs on the different portions of the relay cell dendrites. So because these qualitative observations were strongly suggested, we thought that we can actually now address the question that if certain input clusters, inputs cluster together to form microcircuits to refine the relay cells output. For this, we also utilize another modeling approach called the network analysis, which is really nifty, that can tell you that the probability of any terminal category to co-occur with itself or with any other category on any kind of a different subpopulations we can see. So I'm going to go over quickly these and skip describing this analysis, but I will tell you what the results, what they suggested to us. What we learned from this analysis is that X-type relay cells show a very good segregation of their inputs. There are cortical and retinal synapses are completely segregated on their tertiary for the cortical and for the proximal, that is primary and secondary dendrite branches for retinal inputs. In contrast, non-X-type relay cells show no segregation of retinal and cortical synapses. They were all together. There was no selectivity, and they received very few cortical feedback, even in the tertiary branches. And they displayed atterantia, that is gap junction-like contacts with each other. That was really interesting. So, here's that. So next, I would like to tell you about about animal models that are used to study synaptic plasticity in the sensory systems. And I'm going to show you a few of our studies on this domain. There is a background, let me tell you this, that there is an interesting condition in ophthalmology, and this is how this plasticity field is started basically. The condition called unilateral neonatal cataract. It is rather prevalent and accounts for about up to 20% of all blindness in children. If the condition is corrected before 4 months of age, it's fine. But the correction, which is kind of the removal of the cataract, very simple process. But if you delay it after 6 months of age, there is a considerable loss of equity and a poor depth of perception. So the fact that the corrections before 4 months lead to minimal visual loss tells us that this actually brain that is very actually plastic during this period. And this is actually a lot of the recommendations for the clinic came out for the animal models that actually mimic this condition. So the animal model of the unilateral cataract is generated by closing the eyelid, one of the eyelid shut in a paradigm called monocular deprivation. And this was again used by people and Weasel in the early 60s. What they found was a permanent change in brain architecture. The connectivity patterns in the visual pathways and in the ocular columns have changed. This is demonstrated by placing a transsynaptic anterograde tracer in one of the eyes, and say in the normal eye, and they can do it in the normal animals and monocularly deprived cats and monkeys, and all sorts of other species as well. In normal animals, the tracer travels through the LGN and visualize when they arrive to cortex, it visualizes the stripes on the tangential sections of the cortex. And here we get to see the LGN in this eye-specific lamination here. But in the cortex, these stripes are in the layer 4, and you best see it in these tangential sections. The bright fields stripes are the label that coming from the injection which was put into the one of the eyes. So when you do the same thing in a monocular deprived animals, the axons that come from the deprived eye and the ocular dominant columns they make up, they become smaller, they shrink. This kind of a darker lines here, the columns here. In contrast, the non-deprived eye axons and the columns they make, they expand. They become larger. And just like in human infants, there is a critical period to compensate for the effects of the separation. That is that if you open the eyes before the end of that critical period, the cortical development occurs normally. So there is that critical period is the period during which the brain is still plastic. So we studied this phenomenon in neonatal ferrets. It turns out that ferrets are very advantageous animal models because they are born rather immaturely. A lot of the developmental processes that go on prenatally in cats and monkeys and humans, they happen postnatally in the ferret because their eye opening occurs really late. They also have ocular dominance columns because they have frontally located eyes. So we took advantage of these animals' developmental properties, and they studied the morphological and molecular consequences of different sensory experiences in early life. That is, we wanted to know what factors were correlated with the beginning or the end of the critical periods. In particular, we focus on two paradigms. First one is the dark rearing. We first put the newborn ferrets in total darkness until after their natural eye opening around here occurs, and then kept it in the dark and opened it after varying times and looked at their brains at the end of that period. Because dark rearing delays the onset of the critical period, we argued that the development of any factors that are delayed in parallel to the dark rearing would be tied to the critical period, and it would be also delayed. We also use an opposite of this, a light experience paradigm, early eye opening. Because the ferrets have their eyes closed until they're 1 month of age, which is great, they were very handleable. You know, in a couple of months after they were born, we were able to pry open their eyelids and let them expose to a natural light. This is very analogous to what premature babies experience when they are, you know, have to put into neonatal wards in this under light conditions. So what we expected is that this premature light exposure to trigger the experience-dependent developmental processes and will do so prematurely. So to detect the impact of these abnormal visual experiences on the expression of the glutamate receptors that are involved in the synaptic plasticity, we again use immunological microscopy. We quantify the number of synapses. You can see an unlabeled synapse here and a labeled synapse here. And we do that in layer 4, in layer 2, 3. And we also calculated the ratio of synapses that express two different types of NMDA receptors, NR2A containing and NR2B containing receptors. And what we showed was that the change in the timing of the first light exposure impacted the patterns of NMDA receptor subunit development. And these were tied to different aspects of the light exposure. That is, the NR2B receptor expression was tied to the critical period of ocular dominance column plasticity. Its reduction signaled the end of the critical period. On the other hand, the NR2A type receptor is related to the adaptive processes throughout life, and it is bidirectionally regulated with the strength of the light exposure. Interestingly, NR2A and NR2B receptor regulation has been shown to be very common property of the synaptic plasticity, not only in vision and also in other sensory systems, but also for other brain functions that involve plasticity, including memory and addiction. So we also used electron microscopy to reveal the morphological developmental parameters of the thalamocortical axons. And it also showed that nice correlations, but I'm skipping those data. So one last topic I want to mention is, getting low on time, is the caveats of animal models. There are a number of instances where the findings from a disease model did not apply for the human conditions, such as transgenic disease models used for translational neuroscience, or treatments developed by using pharmacologically induced affective disorder models. But for the basic research in sensory system, problem is more about evolutionary homologies. The nervous systems of different species are evolutionarily optimized to survive in different ecological environments. They can survive they live under the ground, like rodents. They live over the trees, the birds, or some monkeys, or under the canopy of the rainforest, or ground animals and lizards. Depending on the prevalent stimuli that are actually present in any environment, the sensory systems of animals that live on those environments may be optimized to that particular niche. For example, burrowing animals that live in tunnels, such as rodents, use their whiskers and their sense of their sense of smell to find their way around the tunnels where they live. And they use their visual system as their last resort. They actually do not rely on it at all. So it's not surprising that mice may have lost many features of the visual system in comparison to primates. So this is also another important issue in studying the taste of sense. Well, contrary to what Disney thinks about it, the rats do not have gourmet taste perception. They can't discriminate many things, and they don't choose their food based on how well they taste. They merely use their taste system to avoid poisons and other bitter things. On top of that, it turns out that there are major differences, peculiarities in how the rodent taste pathways are organized. In human brain, the taste inputs come to brain from tongue via the central nerves 7 and 9 here. And this information is sent to the nucleus of the solitary tract, which sends information to the taste thalamus, VPNP here, which then relays it to taste cortex, which then sends it to amygdala. So great, this is kind of what is expected. But in rodents, the taste input comes to the NTS and then takes a detour and make a synapse in the midbrain. Interestingly, this midbrain nucleus, PBN, sends the taste information directly to the amygdala and put that in an arrangement that to enable the rats to learn which foods to avoid before they get sick after eating them. So this pathway is a feature that is very suitable for the rat's ecological niche. So this pathway difference is in fact very fascinating. It's fascinating from a sensory systems perspective. It is also fascinating from an evolutionary neuroscience perspective. In fact, there is no other sensory system pathway in mammalian species that shows a similar pathway aberration or takes a detour as does the taste pathways do in rodents. So regardless, and because rat has been the species most of the taste research was based on, rat was the first species we studied. Talented undergraduate in the lab placed an anterograde tracer in the brainstem nucleus, and that project to the thalamus and visualize their axons. There was something very peculiar about these axons. They were really large, both in the light microscopy level and at the electron microscopy level. Here is a labeled brainstem terminal in the taste thalamus. You can appreciate how big that is if you compare it to these terminals here. And the interesting thing is that such large terminals are not encountered with any other thalamic structure, even in the vision and the retinal terminals I showed you before. We constructed these large terminals and their targets and demonstrated that parabrachial terminals in the face thalamus almost exclusively contact these proximal dendrite segments as they emerge from the cell body. And with this localization, these terminals can exert strong activation on these relay cells without leaving much room for modification of taste-evoked activity before it's relayed to the cortex. This also indicates that even though they are analogous structures, the information processing in the chemical thalamus can be drastically different for the non-chemical thalamic nuclei. There are some similarities between the taste and non-chemical thalamus. I'm going to go rapidly because I want to show how this circuitry looks like in a primate-like species. So this is the rat. What we have shown is that there are no glomerulus, no triads, no GABA interneurons, no acetylcholine, no VGLT2. That may be because that taste relay cell there's a simple relay function there, but may also be the indication that there are interspecies differences. So best thing to be done was to study a primate brain, but they are not easy to come by. So the next best thing we have was the tree shrew. So tree shrew is are the closest relatives of the primates, and they're similar to the primates in brain anatomy and their phylogenetic proximity to monkeys and humans. In fact, the rodents separated from the tree shrews and the humans about 80 million years prior, 80 million years ago. That there was a long time ago. But they're native to Southeast Asia, and they can be bred in captive colonies, which we have one in our lab. The nice thing is that the tree shrews are avid eaters and drinkers, which turns out an advantage for studying gustatory behavior. So I'll tell you about, I'm running out of time. I'll tell you about one interesting difference between rats on one side and the tree shrews and humans on the other side, and leave anatomy out. As you know, we eat a lot of processed food, which often contains preservatives to increase the shelf life of the item. And sugar in high concentrations is a great preservative, particularly used in ketchup, jelly, and a lot of other sweet stuff. You may now say that, wait, I don't taste a lot of sugar in the ketchup. That's true, but it's because there's another additive added in the ketchup, a sweetness receptor blocker, so that even though you consume the sugar, you don't realize that you're consuming it because you don't taste it. So the food industry puts a lot of research on these types of taste receptor blockers, but the blockers that work on rats don't work on the humans or vice versa. But our tree shrew shows a sensitivity to human sweetness blockers. Lactazole mentioned in this slide is that blocker that in a two-bottle preference test, the tree shrews prefer sucrose over water. But when you put lactazole in the sucrose, they don't realize that it's sweet anymore. And similarly, in tree shrews, but not in rats, tree shrews can't taste the artificial sweetness, such as NutraSweet, and that makes us actually claim that the fisheries make a really good animal model to study primate-like taste processing. So we did that, and I'm going to skip this faster. We revealed the synaptic circuitry in the taste thalamus of the tree shrew, and we show that everything that we did not find in the red thalamus was there in the tree shrew. There were the glomeruli, GABAergic circuitry, the BGLUT2, which is another good marker for the primary inputs, and how they are segregated. The tree shrew taste thalamus was very much like the tree shrew LGN visual thalamus, and also hopefully how the other primate thalamus look like. So I'm going to stop here because I'm running out of my time, and just want to acknowledge the graduate students and undergraduates and other collaborators who actually did these work. And I'm happy to take any questions anytime.
Hello again. So there were a couple of questions that were asked prior to the session. So to start off, one of our participants asked, as an individual develops, what changes occur in their sensory pathways? For patients with neurodegenerative diseases such as Alzheimer's, do they still experience pathway changes? And if they do, what differences can be observed with typical functioning patients?
So very interesting question. So Alzheimer's disease, the kind of conditions that set later in life, there's an incredible amount of research going on, and it looks like the functional losses that happen in Alzheimer's disease, not pathway specific, but it's a kind of a general location specific. That is, we now kind of know that it is a prion-like disease that spreads from the base of the brain and to the frontal part of the brain and then to back. Because most of the sensory cortex are in the back of the brain, is actually senses get to be spared until the last moment. This is in contrast to other conditions that are actually developmental, things that occur in developmental. For example, autism. It is thought to be based on very many small genetic changes, mutations that can actually happen very early, express themselves early in life. And these changes are on the molecules, such as a metabotropic glutamate receptor, which are required, there are really needed elements for normal development of the nervous system. So when you pull out these kind of a really needed elements out of the equation, as a result of these small mutations that may happen in the autism spectrum disorders, development does not happen normally. The sensory system does not happen normally, so that the oversensation, perception, and so on may be the result of that. Yeah, great question.
Okay, so to move on, another participant asked, which model organisms are the best to study the sensory pathway plasticity, and what is the selection criteria for this animal model?
Great question. So, well, I would say that, so a ferret model I have shown you is wonderful because of these developmental properties. That is, they're born really immaturely. You have about a month after birth to be able to manipulate these sensory environment for them, which is very difficult to do even in a monkey, because a lot of, like humans, a lot of these sensory developmental stages occur prenatally while the animal is still in the womb. But having said that, you will notice that great majority of the plasticity work are done in mice because of the access to these animals. They are very easy to breed. And not only that, we can actually manipulate the genetic environment makeup of the mouse, which gives us many opportunities to identify different cell types by markers, putting GFP tags on them, and also being able to specifically activate the different pathways. So it is usually a trade-off, right? You go to mouse to learn everything really deeply, fast and quick, and then you go to this higher mammals and potentially the primates to confirm what you find in the mouse is actually valid for the primates.
To move on to the next question, when we do use ferret models or mouse models, do these pose a challenge when applying the findings to humans? Because we do know that humans are way more complex beings. So when this occurs, when these problems occur, what measures are used to overcome them?
Yeah, so in fact, there's actually really good examples of that. The studies done in the animal models that actually generated major clinical suggestions. Monocular deprivation paradigm that mimic the neonatal cataract. Until that time, until Hubel and Wiesel did the monocular deprivation on cats, the removal of the cataract was often left for year 1, year 3, because it's easier to remove the cataract, right? And that led to a lot of blindness. But when the animal model using the monocular deprivation revealed that it is a issue of balance between the two eyes. It's not only the lack of stimulation, it is lack of stimulation from one eye. It is an unbalanced stimulation. So the one major outcome of the monocular deprivation research was to suggest the clinical clinic to use eye patches on kids, and also even like that has the strabismus, which actually generates this kind of a monocular deprivation kind of issues. So there can be, even though the animal model is not perfect, the mechanics of the paradigms that we can isolate from these models can actually generate valid applications in the human health and treatment.
Thank you. And to move on to our last question, which has to do with more of our senses. Our participant asked, do our different senses such as vision, taste, and smell have different structured sensory pathways? Or is there a difference in how they are processed and form neural connections? Is there, to be specific, is there a specific sense that activates the most sensory pathways and in turn caused the most connectivity in the brain?
So yeah, each sensory pathway has their own path, right? They have their own sensory organ, they have their own thalamic nucleus, they have their own cortical structure. So they have their own pathway. And how much, and the organization within them, and it's actually our work and others have shown that, is actually follow a very structured template, because the synaptic organizational properties we see in the visual thalamus, we also see in the auditory thalamus, we also see in the somatosensory thalamus and the taste thalamus, right? And that includes the, not only where the input, modulatory inputs are coming from, how they're inhibited, and types of neurotransmitter that are used, and everything that seems to be really kind of overlapping. They use the same kind of mechanisms. Why waste a good mechanism if the brain has one good mechanism? So use it on the other systems as well too. Great. So, but what is the dominant sense for any species? It actually seems like it's determined by what is important for the animal. For a mouse living in tunnels, a rat living in tunnels, there's nothing to look at, right? there may be things to smell. It's dark, you know, there's no need for a vision. And everything is like a tunnel, it's small. And for this animal to move, is actually the most efficient sense is their whiskers. They move their whiskers around, and when they touch the walls, they know that they are not hitting the wall, and they kind of very efficiently is actually sense the texture that are around them. So yeah, it is all where the ecological niche for the animals are. Then this some sense becomes more dominant than the other ones in a way.
Thank you so much, Professor Alev. It was a very wonderful lecture from you. I was very delightful to be a part of it, and I'm sure all of our participants were the same. Without further ado, I am giving back the spotlight to Melike. Thank you again.
Thank you so much.
Hello again. Thank you for this beautiful session. Before ending, can we take a picture as a memory, if you don't mind?
Yes, please. I'll want a copy.
Okay, thank you. Participants who are available can also open their cameras. I will wait. Okay, I'm taking the picture.
Great.
Okay, thank you so much.
Thank you, everyone. Thank you for being here on a Sunday evening. Yay.
Thank you. See you. Our next session will start in 10 minutes. So let's have a break for now. Thank you.